So the next speaker is a nice Breton name, Klervia Jaouen, who's going to speak us about isotope perspectives on paleodiets.
So good afternoon. Thank you very much for the invitation. So today I'm going to talk about what isotope told us on the diet during the Paleolithic. So I'm not going to talk only about my research, but also about results that have been obtained now for maybe 40 years. So the Paleolithic actually covers a very long period of time because it starts with the earliest stone tools 3.3 million years ago and only ends like 12.5 thousand years ago. And so during this time period, you had a huge amount of hominins that lived on Earth. And I think about Australopithecus, Neanderthals, modern humans, etc. And so possibly with this diversity of species comes a diversity of diet. And since we applied isotope techniques to investigate Paleolithic diet, we tried to look at the link that it could have with human evolution. And so there's maybe 4 questions that have been at the center of this link between diet and human evolution. The first one was the link between like where the hominins getting their food from forest or open environment, with this idea that nowadays the apes get their food mostly from forest environment, whereas where you will find a lot of early hominins, it looks like the landscape was more a savanna than a forest. There's also the question of the consumption of meat that's linked to another hypothesis that's called the expensive tissue hypothesis. And the idea of this hypothesis is that over the course of human evolution, you had an increased volume of the skull, the brain, and it could be linked to an increased consumption of meat, with the idea that the meat consumption requires a smaller gut than plant consumption, and the guts are quite expensive in terms of energy, and the brain as well. You see it here on this graph where you see the body weight and the basal metabolic rate for the brain, the liver, the guts and other organs, and the muscles. And so the idea is basically if you reduce the energy that's taken by the guts, then you have more available energy to develop bigger brains. There's also a question that's linked to the idea of were the hominins cooking their food or not, and what's the advantage in terms of energy. But we're not going to talk about this today because isotope techniques currently do not provide any new information on that aspect. And finally, there's the question of meat or fish consumption, because it seems that modern humans were, during the Paleolithic, eating quite a lot of fish, but not the other hominins. So is it like something that makes us human or not? It's something that I would like to briefly talk about today. But before going into those questions, I would like to just give the definition of what isotopes are, because it's not always very clear. And so I will take here the example of carbon isotopes. So you see here 3 atoms of carbon, the carbon 12, 13, 14, with electrons and the nucleus that's made of protons and neutrons. And actually all the atoms of carbons have 6 protons. That doesn't change that what makes an atom of carbon. Whereas the number of neutrons can vary. For the carbon 12, you only have 6, this one 7, and this one 8. So basically the carbon 12 is a light isotope because it has less neutrons than the carbon 13 that has 7 neutrons. And when we do stable isotope study, we look at the proportion of heavy and light isotopes in body tissues. And because we are what we eat, the proportion of heavy and light isotopes in our tissue will reflect the one of the food products that we are eating. So again, I'm talking about stable isotopes and not radioactive isotopes like the carbon-14 that will decay over time. So when we conduct this isotope study, we work on fossils, of course, and so we can work on teeth that are usually mineralized and well preserved, and also on bones that contain the same mineral as the dental enamel, the bioptyte, so again preserved for millions of years. Whereas we can also work in the bones on the collagen, because when you have a fresh bone, you will have about 30% of organic matter. But this collagen is not going to be preserved for a very long period of time, especially in arid and tropical environments. But in Europe, you can still analyze it for maybe 100,000 years, depending, of course, on the environmental context. So now I'm going to talk about those 4 main questions that I mentioned in the introduction, and we are going to start with the diet of the early hominin species and try to see if we can already answer to the question, is there a link between diet and evolution? And I'm going to start with this question of were the hominins getting their food from forest or open environment? And for this, we're only going to talk about data that have been obtained on the dental enamel, because the organic matter and the bones are usually not really well preserved. And I'm going to talk about carbon isotopes. So maybe, I don't know if you know about the photosynthesis, different type of metabolism that plant can have, but you have a very simple example here where we have a tree that in Africa is often a C3 plant, meaning that when the plant is doing the photosynthesis, the first molecule that will be formed will have 3 atoms of carbon. Whereas for grasses, usually the first molecule we have 4 atoms of carbon. So we talk about C3 plants and CO4 plants. And this metabolism will impact how the plant is going to select heavy and light isotopes coming from the CO2, coming from the air. And we can express this selection with the delta values that just reflect the proportion of heavy and light isotopes. So if it's low, it means there's a lot of light isotopes, and if it's high, there's less light isotopes. And so just looking at the isotopic composition for carbon, the delta value of the hominin teeth, we can have an idea if they were getting their food from an environment with a lot of grasses, so probably savannas, or a lot of trees, so probably the forest. And so those are data that have been conducted for quite a long time now, maybe since the 80s. And you have here a summary that has been done like 7 years ago with many early hominins from different parts of Africa, different contexts. And you can see here the carbon isotopic values. And the low values will correspond then to the forest environment, and those ones to savanna environment. So you can see that there's quite a lot of variation. And actually that some hominids that were bipedal were apparently getting their food from the forest environment. So already it answers maybe to the question, is the bipedalism related to a certain type of environment? Not necessarily. And what those carbon isotope studies have been showing is the fact that also sometimes you have species that are closely related, like Paranthropus robustus and Boisei, but they do not have the same adaptation. So the phylogeny is not really related to the adaptation according to carbon isotopes. And finally, what it showed is that still progressively over time, what we see in Africa with different timing is a shift from forest environment towards savanna environment in general. And recently, there was a new type of isotope studies that has been conducted, the calcium isotope study. So we're not talking about carbon isotopes. It's pretty new. You can see like this publication is from this year. And we don't really actually know precisely what high calcium isotope values or low calcium isotope ratio mean. But what the researcher saw is that for early hominins, you had some groups that were having value overlapping with the associated fauna, the herbivores and carnivores from the same site. But again, like for common isotopes, the Paranthropus boisei was sticking out, very different. So probably having a very different ecological niche than the other mammals from the same environment. And also what they saw is that the early Homo lineages had lower value sometimes. They were overlapping with the other hominids, but they could have the lowest calcium isotopic ratios from the assemblage, which could also be seen in carnivores. And so the author hypothesized that maybe they were eating meat. And that's very interesting because it goes along what I was showing earlier, this question of the expensive tissue hypothesis, saying that over the course of evolution, there was an increased consumption of meat. And my research is actually focusing on that aspect. So I work with zinc isotopes, and we're very interested in these isotopes because the isotopic composition can be analyzed in dental enamel that can be preserved for millions of years, and we can definitely see differences between carnivores that will have low zinc isotopic ratios and herbivores that will have elevated zinc isotope ratios. And so we first saw this pattern in South African and Kenyan modern food webs, and then we saw them again in this time Pleistocene food webs, so quite ancient ones, fossil ones from Europe and Asia. So I will just show you the example of a work that has been performed by my PhD student, Nicolas Bourgon. And he analyzed some teeth coming from carnivores, omnivores, and herbivores from Laos, from Maklot, so Pleistocene teeth. And what he could see is that the carnivores, again, have lower zinc isotopic ratios than the herbivores. And what's really interesting here as well is that we had some primates in the assemblage. And you can see the orangutans being in the omnivore group, but this is kind of a debate because many people will say that orangutans are actually herbivores, and you can see that they are actually overlapping with the herbivore range, whereas we also had some macaques in this assemblage, and in Laos we find some species of macaques that are eating quite a lot of meat and a lot of insects, so animal products, and the macaques were having significantly lower zinc isotopic ratios than, for example, the orangutan. So there's really good hopes that we could, with zinc isotopes, trace the proportion of meat in our diet. And we also obtained some first results on humans that were pretty encouraging, where we tried to see first on historical populations so that we don't destroy precious teeth. And we looked at populations that had a diet that was rich in meat, so modern ones or medieval, early modern period ones, and also some populations, so all in France, some populations that were having a diet that was rich in plants, in cereals, so usually coming from a poor context. And what we saw is that, again, the population that had a plant-based diet were having values that were higher than the populations that were eating a big proportion of meat or fish. And we started to apply this to some prehistorical context. So I'm not going to be able to show you the results, unfortunately, because everything was not published yet. But for the most recent one that we did, we have a population from Brazil of hunter-gatherer, and you can see that they had a diet that we think was more plant-based according to archaeological data, which overlaps with our meat-based population from France. But that can be easily explained by the fact that they were getting their food from forest environments, and we saw that also on herbivores, that herbivores for zinc from open environment were always exhibiting higher zinc isotopic ratios than forest environment, which means basically, if you want to interpret the diet, you analyze the associated fauna to the humans, and you have the baseline to interpret properly the proportion of meat in the diet. But what's also interesting with zinc isotopes is that it can trace the age at weaning. So it's a recent result that we just published that we obtained on the hunter-gatherer from Brazil and also the historical population from France. And you can see an example here with teeth coming from 2 individuals, and we have the teeth that are formed in utero that record the signature of the body of the baby when it was still in the womb. And the values are lower than the teeth that are forming during the breastfeeding period. So you have an increase, like the ratio will start to be more enriched in heavy isotopes. And then when you have the introduction of the solid food, progressively the ratio will go lower and lower and reach like this kind of 0.3 value. So just looking at this pattern, we can have an idea at which age the solid food was introduced in the diet of this individual or at the scale of the population, which is interesting because this process is highly cultural, so we could see some cultural variation. And also it has an impact on the time between pregnancies. But there's the zinc isotopes that can provide this kind of information, but more recently it was also shown that calcium isotopes could document that, and it has been already conducted on early hominin species, and they showed that early Homo were actually having a longer breastfeeding period than Paranthropus and Australopithecus. So to conclude this first part of my talk, I just wanted to summarize that currently what we know from the isotopes is the fact that the ecological niche seems not to be related to the phylogeny or the bipedalism thanks to carbon isotopes, and that the expansive tissue hypothesis is now, we can now test it. So like for the research we just saw on genetics or proteomics, I think in the coming years we are going to have a lot of interesting results. And now I'm going to move in time and go to a more recent period of the Paleolithic, because I'm going to present data that have been obtained in the organic matter of the bones, so in the collagen. So we're only going to cover like the last 100,000 years and only for European context where the collagen is preserved. And for this context, we can talk about Neanderthals and modern human diets. And we can also work with another tracer, the nitrogen isotopes, that are used since the 80s. And so we kind of really know them well now. And you can see in this graph what type of information they provide. So basically, we usually do carbon and nitrogen isotopes together. Carbon isotopes in Europe will be different between marine environment and terrestrial or freshwater environment. And the nitrogen isotopes will provide an information on the position in the food web, the trophic level. So basically, the higher the nitrogen isotope values are, the higher you are in the food web. So you can see plants, herbivores, carnivores, and the fish will have the same values than some carnivores. And so let's see now where the Neanderthals and the modern humans are plotting. And usually for the Paleolithic, this is what you see. The Neanderthals usually have slightly higher values than the carnivores, which has been interpreted as the fact that they were simply top-level carnivores. And the modern humans were having even higher nitrogen isotope values, which has been interpreted as the sign of frequent fish consumption. And so with this came the idea that maybe fish consumption is something that's typically human and that it was not done by Neanderthals. But a few years ago, we analyzed the collagen of a Neanderthal, and we realized that the value was plotting with values that we usually see for modern humans, and so which would be usually interpreted as fish consumption. And at the same time, another team saw modern humans values that were super, super high for nitrogen isotopes in a context, in an archaeological context, where you didn't have a lot of fish remains. So we started to question our interpretation. And actually, we have, since a few years now, a new type of technique where we go to the molecular level and can look at the trophic level with more details. So it means that we are not going to analyze the isotopic composition of the collagen as a whole, the bulk collagen, but we are going to look at the isotopic composition of the amino acids taken separately, so the amino acids that form the collagen. And so we can, for example, look at the nitrogen isotopic composition of the phenylalanine, which is not impacted by the trophic level, but other factors, such as environmental or metabolic factors. Whereas the glutamine, actually it's also impacted by the same environmental or other factors than the phenylalanine, but also the trophic level, which means that if we take the same trophic level, the value will correlate, like they vary the same way for one specific trophic level. But if we change the trophic level, we just simply change the line. And so we can see, for one side, all the herbivores on the line, all the carnivores on the line, and the fishes on another line. So with this, we just need to analyze the collagen of a Neanderthal, and we know if it was carnivore, herbivore, or a fish eater. And so here are the results for 2 studies that have been conducted by a team in Tübingen and in Japan and our team in Leipzig. And so you can see, I will start with the work that we did with the Neanderthal that had the super high nitrogen isotopic value. And you can see this Neanderthal here plotting exactly on the carnivore line, which means we do not explain this high nitrogen isotopic value by the consumption of fish, but more the consumption of herbivores that themselves already had high nitrogen isotopic values. And now you can look at the Neanderthals from the Spee Cave, and they are plotting just below the carnivore line, and they were exhibiting quite classic nitrogen isotopic values. So they do not plot exactly on the carnivore line, but it seems that they still had a diet that was pretty rich in meat. So this goes with the interpretation that we were making for years on the diet of Neanderthals with isotopes. And now, what about those modern humans that we saw that were having very elevated nitrogen isotopic ratios? So they come from Burankaya, which is one of the sites with the oldest human occupation in Europe. And I'm going to present right away the results for those humans. And you can see the herbivores values that are plotting close to the herbivore line, so it seems to be all right. The wolf is on the carnivore line, and you can see here some foxes that are interpreted as omnivore foxes, and the humans. And so you can see that the humans are first plotting below the carnivore line, so it seems that they had a diet, quite an omnivore diet, but also it's very far from the lines that would have testified from fish consumption. So a value that we would usually interpret as intense fish consumption is actually apparently no fish consumption or not frequent. And what you can see here is just below the humans on the herbivore line is a mammoth. And so the mammoths usually have elevated nitrogen isotope ratio, and this is what would explain such high nitrogen isotopic ratios in their consumer, so meaning that those humans were hunting and eating mammoths. So in conclusion, what we can say is that the paleo diet and the study of paleo diet with isotopes is not something new because we now do it for 40 years, but it's our understanding of those diets through isotopes is currently fully changed by new isotope technique. So zinc isotope, calcium isotopes, we are now able to measure almost all the periodic table for isotopes, and with this come new information. And we're also able now to go to the molecular level when we do those amino acid isotope analysis. And with those analyses, we realized that we first thought that neanderthals and modern humans were having distinct diets, and with those new techniques, it seems more complicated than what we thought, and maybe we were wrong. So I think a lot of things are going to come in the next years with interesting results, and I hope that we will see this very soon. So I thank you for your attention. I would like also to thank all the researchers that have been contributed to the zinc research and amino acid research that I presented, and the funding agencies. Thank you very much.